Hello there. It's been a while since I've been on. How are you guys? Are you enjoying the weather? Because I'm dying in this heat. After all that rain and then the heat just comes on intensely. Thank you for joining Autism Belize tonight. We have a very special presentation. We've got two amazing PhD candidates from McGill University. Nuwan, who studied genetics, and then we have Danielle Almeida, who does the neuroscience part. All new to me, but I am super excited because so many times when I give talk about autism or I talk to parents who are just going through the diagnosis, we have so many questions about why. You know, even as Mateo grows up, I still get more questions. You know, as he's heading into teenage years, and now we're dealing with anxiety and depression and a whole slew of other things that are just beyond the autism thing. And the more you read, you find out, yes, you get the autism diagnosis, but then you have sensory processing disorders, and you have this, and you have that. And the link between and the reasons why, it's still such a puzzle. It's one of the most researched things that they're studying all over the world right now. And these two young men are doing like really cutting edge research to study neurodevelopmental disorders and the brain and the genetics behind it. And I'm really just very interested to hear what they have to say. I've never been a science person, so I might have to like take notes and ask really silly questions. So bear with me because this wasn't my field ever. But I feel like for all of us out there, whether we're parents or professionals working in the field, it's important for us to know what's being studied, what's going on, what are they considering between links. And I say professionals because I feel like this is going to benefit mental health professionals, pediatricians, neurologists. I know you guys have studied and you know your thing, but every day there's something new coming up, and these are what they're working on. Might be huge breakthroughs in a couple years that might affect all of us, hopefully, in a great way. So I have one presenter already on. The next one is having a little bit of technical difficulties. So if you could bear with me, I will wait just a little bit. I know we've got about 7 people on already. So we're gonna get started very shortly. We're just having a problem with the video camera because we hear the mic, but not the video. So we'll just give them a little bit of time. For those of you who are just signing in and haven't been to one of our live presentations before, the guest speaker will come up, they will present. You can post questions on the side in the comments bar on your Facebook page. These presenters have asked it, they said they will answer your questions after the fact because they've got a whole slide thing to do. So post them, and as soon as they're done with whatever, we'll start addressing as many questions as we can. If we can't do it in the time that's allotted, we can go back afterwards and just type in your answers there. This is probably our 12th or 13th broadcast. It is slightly different because usually we talking about different therapies or hands-on tips for parents. While this is definitely more it's on a different level of education. So I'm hoping we'll have a wider variety of people watching. If you know someone who might be interested and who missed it tonight, we will have it up on our website. That's autismbelize.org. And usually it's up between 24 hours. You just need to go to autismbelize.org, go to speaker series, and all of our presentations are there. You click, it'll come on, and you can watch the thing in the entirety. Unfortunately, you would have missed the opportunity to ask questions, but you can still go and learn. And I feel like this is something that should be shared all around. It'll be public, so you can download, share, encourage your class who's studying chemistry or biology or anything like that might be interested to do this, okay? We'll go on, and I'm just gonna have Daniel come on so we can chat a little bit and add him to the screen. Everybody, this is Danielle Almeida.
Hi, thanks for having me.
Daniel, where are you?
I'm in Montreal right now, and the weather's pretty timid here, so I'm enjoying that.
That's great. We are very, very hot. Oh, I did the calculation because you were talking to me in Celsius. So right now is after 6 o'clock, and today wasn't our hottest day, but at this point in time, it feels like 37.2 degrees Celsius with humidity.
Wow.
Yes, we are definitely going through a heat wave, which is kind of scary with hurricane season, but we're going to keep our fingers crossed that we're protected this year after everything that's happened around the world. So I want to thank you for coming on and offering to do this for us while we wait for, oh, I see him. He's back on, but still no video. We're working on it for another guest speaker. Can you tell me a little bit about yourself?
Yeah, sure. So my training is as a molecular neuroscientist. So I use post-mortem human brain samples to understand the biology of psychiatric disorders. And a lot of post-mortem researchers, sorry?
Psychiatric disorders such as?
Depression, suicide, the impacts of childhood abuse. And a lot of researchers also look at post-mortem human brain samples of individuals who suffered with autism. And so that's the sort of the approach that I'll be taking, is looking at the post-mortem human brain research.
Wow. Okay. Okay. So I know we're already have questions, but we're going to wait till they present. Okay, we're still having some technical difficulties. How confusing will it be if we let you go first?
I think I can go first. Yeah, I think that's totally fine. Nuwan will be presenting on the genetic side of things, and I was going to build off of that and look at the neuroscience side of things, but I think that I think that they're pretty standalone. So we can go ahead and start with my presentation if you'd like.
Okay. Well, I'm going to give you the screen and get myself off here.
Okay. Can you just let me know if you can see my screen?
Here. We can do it.
Okay.
I see him. I see him. I see him. Okay, wait. Hold on. I'm getting excited. It's okay. Let's see. Nuwan!
There you go.
Hey, when I was in school, the science guys were not this hot. I've just got to say. Oh, missed out on the revolution there. Thank you so much for coming on Autism Belize and doing this for us. Everybody's been excited about this presentation. So we are going to let you introduce yourself, and then I'll let you have the screen. Do you have any questions before I kick myself off?
No, I think I finally figured out these technical difficulties. So
you're on. I see the mic. We've got lights. When you want to share your slides or anything, there's a share screen button down there. You click it. I'll be monitoring, and I can also put it up. So I'll have your picture and the shared screen.
All right, just want to confirm, can you all hear me clearly?
Perfectly.
Perfect. All right.
We hear you. We're ready to go.
Perfect. So just to introduce myself very briefly, I'm a PhD candidate at McGill University in the Department of Human Genetics. So what this means is that I study rare and severe neurodevelopmental disorders. So these are disorders that are characterized by severe mutations, and obviously, as the name implies, very rare in the population. So I'll talk a bit about what I do in the following presentation, but essentially we combine a host of genetic technologies to try and get to the root cause of how these disorders come to be and how they lead to debilitating symptoms in these children who have these severe disorders. So jumping right into it, I'll share my screen. All right. So just want to clarify, is this visible to everyone? Okay. So, okay, perfect. All right, so thank you again for having me on and for having the opportunity to discuss some of the research that I do in the context of genetics and autism spectrum disorders. So like I said, I'm a PhD candidate at McGill, and I'll be discussing some of the work that we've been doing thus far, especially in my academic career. So jumping right into it, neurodevelopmental disorders, it's quite a mouthful to say very quickly, but NDDs, as I like to call them, are a heterogeneous group of conditions that are essentially characterized as this cluster of conditions that have very similar but different phenotypes or symptoms. So at the top here, I very strongly emphasize that autism spectrum disorders are just a single category of this broad spectrum of NDDs. However, NDDs can also encompass global developmental delay, some social communication disorders, intellectual disability, and ADHD. So there's quite a lot of variability in how these NDDs present themselves. So no child with an NDD is identical to another. Essentially, there's a whole host of symptoms, a whole host of variable phenotypes and conditions that characterize someone with a NDD. So obviously this is Autism Belize, so people are well aware of what autism spectrum disorders are. But ASDs generally range on a spectrum, as the name suggests. So we have on the one end, high functioning autism or level 1 autism, in which children or individuals require support but very little. These children often require some sort of behavioral training, more or less, depending on the severity. But these individuals have difficulty initiating social interactions, might have some trouble with communication, but overall they're able to function in society as we see as normal, and function such that some people might not even be aware that these individuals have an autism spectrum disorder. Now moving on to level 2, which is the more general feature of autism that most people are aware of. These individuals or children require substantial support in their day-to-day activities. And again, because this is autism, it's marked by severe or mild or moderate, depending on how you fall on the spectrum, deficits in social interactions. So these children might have difficulty, initiating eye contact or communicating with others around them. And then what I want to focus on for the rest of my talk is this severe autism, which is on the more extreme end of the spectrum, which is level 3 autism, if you want to categorize it that way, in which these children require substantial support. So this means help with feeding, walking, going to the bathroom, just general lifestyle requirements in which the parents or other caregivers have to continuously care for the child and help them with their daily tasks. So these are often comorbid or associated with intellectual disability or cognitive impairment, which is quite severe and quite debilitating, as you can imagine, for children with these NDDs, or ASDs, as you would like to categorize them. So what I would like to emphasize here is that autism is primarily a genetic condition. So what this means is that there's a large component of the onset of autism that is attributed to genetics. So while we don't have an autism gene, so to speak, there's no single known gene that causes autism, but we know that there are some genetic factors that may contribute or do contribute to the onset of an autism spectrum disorder or an ASD. However, because this is not 100%, we know that there is an environmental component to the occurrence of autism spectrum disorders. So these, for example, include the age of the parents at the time of conception, any complications during birth, and of course, any sort of family history with autism that may be running in these families. So people have historically increased their knowledge on the genetics of autism, and we've identified, at least according to what I've seen recently, at least 100 genetic variants that are associated with autism spectrum disorders. What this means is that there are many genes and many variations of these genes that are out there that affect the prevalence of autism or may increase your risk of developing autism. However, these are generally variants of small effect. I'll explain this a bit more downstream. However, this sort of means that we all have some sort of mutations within us, in all of us, that might confer a little bit of risk for autism. However, it might not be enough to show on the autism spectrum disorder. So to put a number to what this genetic liability looks like, some scientists approximate that there is approximately 60% heritability or a liability of a genetic component to developing autism. So it's at least a quite significant chunk of information that kind of tells us why autism occurs and how it occurs. So obviously this is a very complicated topic that might not be discussed quite frequently within the autism community. However, understanding your DNA is quite important, in my opinion, to understand how autism is inherited or how it just appears in these individuals without any family history. So, of course, we're all aware that there are different cells throughout our body. There are skin cells, hair cells, brain cells, for example, and all these cells contain a small portion of genetic material that is packed into what we know as DNA. So DNA is, we've all heard of this before, but it's a small, tightly wound double helix structure that contains a lot of the necessary information that a cell needs to develop and to run its normal functions in a human body. So we have DNA, and then we have chromosomes, which are tightly compacted forms of DNA. So all of us have chromosomes that we receive from our mother and our father. Approximately 50% of this is from either parent. And so from these chromosomes, it's where we inherit our genetic material, which ultimately defines not all, but a lot of how our overall physiology manifests itself. So it's not easy to give a full background on genetics within this short time span, but what I want to emphasize here is that these DNA molecules contain something called nucleotides, which very simply can be categorized as either a T, an A, a G, or a C. So we have a variable, massive combination of different sequences of these letters, which is kind of like a sentence in a book. And the book is what you would call the entire genome that is within all of us. So these are the individual letters that define our genetics and help understand how our cells behave and function in a human being. So again, very briefly, a gene, for those of you who are not familiar with what a gene is basically a stretch of these sequences or these nucleotides, these A's, T's, G's, and C's. So the structure of a gene is, for simplicity's sake, just a stretch of DNA here, where you have something called an exon and an intron. Introns generally are found in between exons and are generally taken out as these sequences or sentences, as I like to call them, are read and given instructions to how a cell should behave. So the exons here are quite important. I'll talk about them a bit later, but exons are essentially what forms the basis of a mature gene as it is expressed in our bodies and in our cells. So like I mentioned, within a chromosome, we have different genes that are present and are expressed at different times depending on which cell type you're looking at and depending on a host of other factors that we're beginning to understand more and more each day. So for example, on a specific chromosome, we might have a single gene that is found there, which may code for color blindness, for example. So we see color blindness as something that's controlled by one gene. So we call this monogenic, mono meaning one, genic for gene, so one gene. So we see color blindness as a trait that is often associated with a single gene. And then this gene is expressed depending on the variant that you have, so that the individual who inherits this gene either has color blindness or doesn't, depending on what you have. This is also present in another trait. So these earlobes that we have, they can either be attached or free floating or non-attached, as I like to say. This is also dependent on a single gene that we've identified on a specific chromosome. So what we need to understand is the DNA that we inherit or that we don't inherit from our parents ultimately affects our overall neurodevelopment. So how we develop our central nervous system as we progress from being an embryo to a full-grown adult. So you may see seemingly healthy individuals who have children with severe neurodevelopmental disorders or severe autism. And you may be wondering, how is this possible when the parents are both healthy and they don't appear to have any sort of genetic mutation? What we found is quite often children who are developing these severe or rare NDDs often have mutations that appear out of nowhere. So we generally classify these as de novo mutations, in which there is no inheritance of this genetic mutation. Just as these genes are passed on from their parents to the child, something might have gone wrong where part of the DNA might have been changed or mutated. So genetic mutations are pretty common, and they're normal. We shouldn't be afraid of them. Genetic mutations happen every time a child is born. So no child is exactly 50-50 of their parents. There's often many mutations that are seemingly innocuous and have no effect on us. But sometimes, unfortunately, these mutations that occur have a very strong effect on how neurodevelopment occurs. So this is also present or seen in other conditions. For example, schizophrenia is an example of a neurodevelopmental syndrome that includes a very strong genetic component, but for some reason it seems to occur in individuals who may not have parents with schizophrenia. As you can imagine, a lot of individuals who develop schizophrenia may not be able to have children because of the lifestyle choices that they are forced to make, or the psychosis, or the myriad of symptoms that they experience. So it's not necessary that individuals with a disorder pass on those traits or genes to a child, and then this propagates this phenotype in this syndrome. It's very common where we see mutations pop up in the population that may lead someone to developing this severe syndrome. So essentially what I want to get at here is that inheritance is largely boiled down to statistical probability. So we all have genes and different variations of these genes that can be passed on to our children, but how they are passed on and in what combination is largely attributed to probability. So I talked a lot about mutations, genetic mutations, and how they may be beneficial, how they may be inconsequential, so we see no effect of these mutations, or they could be quite harmful. So I just want to explain to those who are not aware or not familiar with genetic mutations what kind of mutations may occur in an individual. So I typically use the sentence, "the cat ate the rat," to kind of explain how genetics works at a very simple level. So if you all recall, I mentioned the A, G, C, T nucleotides, which are the letters or the part of the alphabet that forms the DNA in our bodies and tells our cells what to do and when to do them. So for example, we'll take the sentence, "the cat ate the rat" here as a stretch of DNA, in which each word and each letter is quite important to give an instruction to the cell. So for a silent mutation, as the name suggests, has probably no effect that we can readily observe. A mutation here would be, for example, the C would be replaced with a K. So in this case, you can still read the sentence as "the cat ate the rat." The spelling might be off, but the sentence meaning and the understanding is still there. So similarly, in a cell, if they were to read the sentence, they would still be able to understand the instructions that are presented by the DNA there. On the other hand, we have something that might be called a substitution mutation, where the letter might be changed into something else that may alter the meaning of the sentence, at least slightly, or depending on the mutation, quite markedly. So in this case, the cat ate the rat was changed to the hat ate the rat. So obviously, this sentence is technically grammatically correct. However, it doesn't really make sense that a hat would eat a rat, as you can imagine. So while the sentence does make sense, and there is a clear instruction there, the fact that cat was changed to hat greatly changes the meaning or the definition of the sentence. So it creates a different set of instructions in the cell, which, as you can imagine, might have some severe effect. And something a little more straightforward is when there's a deletion. So in this case, the T in the cat is deleted here. So instead of the cat ate the rat, you would see the ka tet her at. So obviously this is nonsensical and probably doesn't make sense to anyone who was to read the sentence. And as you can imagine, in a cell, if it were to receive DNA, which was deleted at least by more than one nucleotide or letter, this stretch of DNA might be nonsensical and would create a host of problems in the cell in terms of how development occurs. The same is true when there's a letter inserted. So instead of the cat ate the rat, you see the ika tat ep era t, which doesn't make sense at all, of course. And again, similar to deletions, this changes the sentence structure quite dramatically and makes it kind of incomprehensible for a cell to be able to express this gene correctly. And something that is also interesting is a nonsense mutation, in which the sentence is abruptly stopped. So we can imagine this in English language as a period being added. So there are some letters in the DNA code that code for when a DNA should be stopped and should stop being read by a cell. And these are generally very, very carefully placed such that the DNA sentence structure is accurate and proper. However, there might be mutations that introduce the stop prematurely. So instead of the cat ate the rat, it could just be the cat ate. So the sentence is there, but quite a bit of information has been cut off, thereby reducing the amount of information that the DNA provides a cell in order to express its genes and to function properly. So what I want to mention here is that new genetic factors are continuously being linked to autism. What this means is that there are new studies, new technologies being developed that are consistently trying to identify rare or common mutations that may affect the onset of autism spectrum disorders. So what some researchers often look at are something called single nucleotide polymorphisms, or we generally like to call them SNPs. So a SNP is just a very simple change in the letter of a specific stretch of DNA. So one individual might have an A at the specific position, but your neighbor or your family member might have a G in that position instead. These are actually quite common throughout the genome and through many of our genes. Most often they're innocuous. However, we find that these SNPs might have a large contribution to how autism develops, especially when there's an accumulation of harmful or deleterious SNPs. On the other hand, we look at something more direct, where we just sequence all the DNA in our cells. And I mentioned briefly before exomes, which are the major instructors of how a gene is formed and expressed. So individuals or researchers might choose to look at only the exons in their DNA, or they might choose to look at the entire genome, the entirety of our DNA in total, to try and find or pinpoint genetic variations that might predispose or increase the probability that someone might develop a severe or rare autism spectrum disorder. So I'd like to just kind of summarize what I described earlier in the fact that high-level functioning autism is generally more mild, and these are often seen as something called polygenic. So they're usually milder symptoms that have a genetic component, have multiple genes that are involved in the syndrome. So this means that there are multiple genes that have variations, but a very small effect that might not on its own create a disorder or any sort of issues. But again, when accumulating these multiple low effect mutations, then there might be some sort of phenotype or syndrome that occurs as a result. And then on the other end of the spectrum, where you have very severe autism, which is what I generally study, these disorders are usually quite rare because these mutations are quite severe and are not inherited, like I mentioned, as people who have these disorders may have a tough time reproducing or finding individuals who are able to have children with them. So these are disorders that are generally characterized by a mutation in a single gene. For example, FOXG1, MECP2, mutations in these genes. Just a single mutation may lead to Rett syndrome, which is a very common NDD. So often these rare NDDs or ASDs that I study have a whole host of symptoms. So not only neurodevelopmental issues, but issues with their motor abilities or their kidney, their heart, their bone structure. There are a lot of phenotypes or symptoms that are associated with these rare neurodevelopmental disorders. We find obviously that cognitive ability is severely impaired, and that is generally what we focus on, such that the brain structure might be impaired, brain sizes might be enlarged or quite small compared to normal. Epilepsy may be common, things like that. But we also have situations where a child is not able to eat or able to make eye contact with their parents or communicate with their parents at all. So there's a whole host of symptoms that are attributed to these rare disorders. And like I said, no child is exactly the same. Two children might have the exact same mutation in the same gene, but it would be quite surprising to find if they would have the same symptoms. So we like to look at these individuals as what they are, individuals, and try and understand how their specific mutations might contribute to their disease onset. So because it is so difficult to identify a specific diagnosis for someone with a rare neurodevelopmental disorder, like I mentioned, there's a whole host of symptoms that are associated with these NDDs, and many of them are present in many different types of NDDs. So you may see one child with one mutation in a different gene who might look almost identical in terms of symptoms to someone with a different mutation. So how do clinicians or even parents try and understand why their child has this severe, rare neurodevelopmental disorder? What we routinely promote and what clinics are trying to promote now is genetic testing. So what this means is you can take a sample of DNA from your child and run a series of genetic tests. So this might include something called karyotyping, where we look at the chromosomes in your child and see if there are any structural abnormalities in how these chromosomes are shaped, if any chunks are missing, if anything is deleted, moved around, things like that. We also perform generally chromosomal microarrays, which are basically this plate where you can run your DNA samples and see, similar to karyotypes, if there are any major chromosomal abnormalities that occur. In general, when we do see these chromosomal abnormalities, we typically find that these are usually the reasons for why children develop these severe neurodevelopmental disorders. But when these obvious tests are done and we aren't able to find anything, we also try exome sequencing. So like I mentioned before, sequencing only exomes of a gene, of all genes, or just looking at the DNA in its entirety in the child and seeing whether or not we can pinpoint any mutations that may be attributed to the disease in this case. So it's quite difficult to diagnose or detect autism, obviously, through a genetic test. We're finding that it's more possible now with advancements like technology, but this is an ongoing process. And so a negative result does not necessarily mean that there's no mutation in an individual. It just could mean that we haven't identified a gene that is attributed to this phenotype or this feature. However, finding a genetic cause for these severe disorders is often, at least we find, very relieving for parents who are unable to figure out why their child has this severe disability and why none of the treatments thus far have worked. So obviously, I mentioned a lot of genes have been identified, a lot of different variations of genes have been identified, and we're finding new ones every day. But this is just to represent that there's a whole host of genes out there, which any sort of variation may, not definitively, but may introduce some sort of autistic feature or neurodevelopmental feature in a child. So unfortunately, because these are rare disorders, there's little attention given to them. They're very severe, and oftentimes most children are very debilitated and not able to be cared for quite effectively because not much is known about these disorders. So we often rely as researchers on the families themselves to fundraise for us and to provide us with the financial resources to study these mutations and these disorders. So what is quite important for understanding how these rare disorders function and come about, what we generally use is models, essentially. So genetic models for the disorder. What this means is that we're able to recapitulate or simulate the autistic or NDD features in a mouse or human cells. So we also, generally people tend to use mice as models for these severe disorders. More recently, we've been using macaque, which are non-human primates who are obviously more closely resembling humans. And in our lab, we routinely use stem cells. So stem cells from an individual who has the mutation. We're able to examine their own cells in a dish to be able to understand what might have gone wrong at the genetic and cellular level to create the onset of these NDDs. So what we normally do in our lab is we ask children with these severe disorders to provide us with either skin, urine, or blood samples. So from this, we're actually able to extract cells that are able to grow quite nicely in a dish, and we're able to culture them or maintain them in incubators, freeze them, and manipulate them for our research. So what we actually routinely do is take a sample of urine from a child, spin it down, and we're able to collect a small portion of cells that are passed through the kidney and grow those to create our stem cells. So by reprogramming or converting these kidney cells in our dishes, we're able to create something called iPSCs, or more generally stem cells, which, for those who are unfamiliar, have the propensity or the ability to become any cell type in the human body. So obviously you can imagine this is quite a powerful tool, as we're able to examine a host of cell types and symptoms that are found in these rare disorders. So I just want to highlight here on the left, this is what a stem cell looks like under a microscope in our dishes. And then because these are very rare, very difficult, and very valuable cell types, we have to make sure that they are true stem cells and that they can become any cell type in the body. So we routinely stain our cells with these fluorescent markers here, as you can see, just to make sure that the cell we're looking at is indeed what we would like to see. So sometimes we can get some pretty pictures here, but essentially what we want to see is that these cells are exactly what we expect them to be. So from a stem cell, like I mentioned, we can create any cell type in the human body, but because we study neurodevelopment, what we do is we prefer to differentiate them or convert them into brain cell types. So what this means is that we take our stem cell and then introduce some chemicals in the dish, and after a few weeks, we're able to slowly differentiate them and convert them into neural cell types, and ultimately neurons that are pretty similar to what you would find in a living human brain. So essentially what we have here is a live replica of a child's neurons growing in a dish that has the same genetic material and is taken directly from the child itself. So obviously without having to go inside someone's brain and pulling out neurons, which is obviously impossible for a living person, we can instead grow their cells and create their own neurons so that we can just look at them in a non-invasive manner. So with this, what we essentially have are neurons that are genetically identical to the source individual. So in this case, a child with a rare neurodevelopmental disorder. And because of this, we're able to study all types of mechanisms or drugs to try and understand what kind of happened or what went wrong in these NDDs. The other cool and interesting thing that we're working on is genetic engineering, where we're able to selectively cut portions of our DNA, replace them, or just leave them as cut to create genetic mutations ourselves. So we routinely do this to create artificial mutations in a healthy strand of DNA to see if we get the same effect in our cells as we do in our patient cells, to try and very tightly pinpoint what is the cause of these mutations on a child's brain cells. So this is the ultimate question that we would like to answer. So we can do this by just, for example, here, changing this G to another letter and creating a mutation ourselves. And we can also do cool things like adding a fluorescent tag to the end of a gene. So we can see this is an example here of a gene called FOXG1, and we added a protein to the end of this gene that expresses the color red. So wherever the gene is expressed, we also see the color red. And this is quite important for a lot of our studies as well. So like I mentioned, these genetic mutations often lead to changes in cell characteristics, which is essentially what we would like to identify. So how does a mutation in a single gene affect the brain cells that we see growing in a dish from a child with these severe disorders? So we like to compare the two, neurons created from a healthy individual compared to neurons created from an individual with a rare NDD, and see if we can find any sort of differences in these cells to understand what might have gone wrong during neurodevelopment from gestation to infancy. So for example, I just want to show here, this is an example of something we found in a syndrome called Actyl 6B syndrome, where we found that these neurons from a healthy individual were growing quite normally compared to an individual with a rare ASD. So that's one example. The other great thing of having these precious cell types from these individuals that we're able to test drugs or medications on these cell types. So obviously, multiple children have tried different medications to try and figure out what might work best for improving their symptoms. But we obviously don't want to just throw drugs at children, hope for the best, and see which one works, which one helps them. Obviously, that is quite detrimental to a child and is quite stressful. So having their own cell types, their own neurons growing in a dish, we can test drugs directly and see if there are any positive changes occurring. So like I said, if we're able to see any issues in patient neurons, we can see if adding a drug is able to reverse those symptoms or those structural changes in these neurons. Also, because we can tag on fluorescent markers to genes, we can find drugs that increase the expression of genes that are mutated to help compensate for the mutation in these children. So what I want to emphasize here at the end is that there are a lot of new discoveries being made all the time, and that we are discovering, for example, new mutations in diseases that have not yet been diagnosable. So Actyl 6B is one example where we discovered a new syndrome. There are new diseases being popped up all the time because of the advancements that we're making in genetic testing. We're also improving our ability for computational analysis. So statistical and computational methodologies have improved significantly, which allows us to scour the genome and the DNA of all these individuals, thousands and thousands of individuals with ASDs, to be able to identify what are the common genetic variants that may lead to their symptoms and their diagnosis. So exome sequencing, DNA sequencing is quite common. And obviously, the ultimate goal is to try and help these children, these individuals with these severe disorders, and hopefully find some sort of drug treatments that may at least slightly improve their overall lifestyle and improve outcomes for them as they grow older, so that they're able to have a more meaningful life as they, mature from infancy. So that is all I want to talk about today. I just want to acknowledge very quickly the university that I work at, McGill, and the Douglas Institute, in which I conduct my research, and our funding institutions that are generously supporting our research as we go forward. Thank you.
Sorry, I forgot to unmute my mic. I have questions.
Sure.
But will you stay around so after Danielle we ask more questions, or should we ask you your questions now and then come back? I don't know.
I guess now is okay. That's fine.
Okay. I don't see any questions yet from there, but I personally have.
Okay.
Okay. You talked about the different. I have a severely autistic level 3. What are the percentages then? Because you were talking about genetics and mutation, but that severe is not inherited. So what sort of percentage would I have if I had another child that child would be on the
Right. So it depends on your child's mutation, for example. I'm not sure if you're aware of if there is a mutation attributed to that condition.
We've participated in sick kids. They've taken a lot of our stuff, but
okay,
I don't know
so far.
Yeah.
Okay. Well, generally what we ask parents to do is if they are concerned about having more children, and whether or not their children might have an autism spectrum disorder, is to get themselves tested as well. So like you've done, we generally like to look at both parents, see if there are any. Yeah. So I'm not sure what's happening on their end, if they're slacking or not, but there are also cases where like you might not see, like I said, any mutations in both parents, and a child just, unfortunately, because of whatever situation, bad luck, or the statistics weren't right, a mutation might just pop up out of nowhere as they were undergoing fertilization. So we often tell parents it's not their fault. These things happen. It's out of our control. You never know when these mutations can occur. So if you are considering having more children, it is important to get yourself tested, but there's always a risk, no matter who you are, to having children with these conditions. So we like to look. Yeah.
Having another child, you talked about doing the genetic testing so you can see everything. If a parent with a child with autism, that's probably, we're not going to be able to do that. Is it worth? Should we try and do that? Get the testing done? You said with the genetic testing, sometimes a negative doesn't necessarily mean anything.
Right.
That some of it can. Is it worth us trying to come up with the money and go somewhere to get this test done? I mean, is that something we recommend to parent with a child?
Yeah. So again, it's all dependent on the family and their circumstances. So there's no clear-cut answer there, unfortunately. But if you are considering, for those of you who are considering having more children and are concerned about having more children with an autism spectrum disorder, I think it is worth investing in getting those tests done if you have the financial resources to do so. So the price.
Just another child, like, is there going to be, if I spend all this money?
Yeah.
Am I gonna have? I mean, they're gonna end up benefiting my child. You're gonna give me a bunch of tests and tell me this and this and this. Can I then use it for therapy or medications and stuff? So I'm just wondering.
Okay.
I don't know.
Yeah. So it depends on the mutation. I'm sorry, this is not a direct answer, but there are some mutations out there that are more well studied, like the Rett syndrome mutation in MECP2. So if that were to pop up in the genetic test, I know there are drugs being actively designed for individuals who have that mutation. But if it's something more rare, you might not see as much research dedicated to developing treatments for that. So we can not promise, but we can try and develop treatments that are tailored to the specific genetic mutation. Because after figuring out what the mutation is, we can find out what effect it has on the cell and how it affects the child's brain. So we can work from there to try and understand how to treat them best.
All right, we have another question that came in. So if you have two children that are autistic, one is severe, one is high functioning, is it inherited, genetic?
That's tough. So in general, if you see more than one child in your family who has an autism spectrum disorder, there's a high likelihood that there is a genetic component. Like I mentioned, siblings might not have the same genes or variations in genes depending on their parents. So it's all probability, right? So if two parents have some sort of genetic risk for autism, one child might get both of those genetic risks. One child might not get any of those genetic risks, and one child might get, some of the risk. So it all depends on probability. But because you see more than one child, it kind of gives you a little hint that there might be something genetic going on there.
I have one last question for you that I don't know if it'll be for you or Daniel. Stem cell therapy.
Right.
They do it in Mexico, they do it in Panama, it's our neighbors. Is that something that you think would benefit someone on the spectrum, especially if they're more on the severe end of it?
Right now, I wouldn't say it's something that would be of benefit. So generally, stem cell therapy is done in more advanced conditions like Alzheimer's and Parkinson's. We start to see clinical trials for those. But for autism spectrum disorders, the research is still in its infancy, and I wouldn't say that's something that can be confidently considered at this time.
All right. Let me see. Right now, we don't have any questions, but I'm going to ask everybody to stay with us because we have the next part of our presentation. Thank you so much, Nuwan. I don't know if you want to hang around just in case we get some more, but we've got a lot of people still watching, and I'll give Daniel the chance to come up. Oh, see, we have one mom that has commented as a child on the autism spectrum. She has done stem cell treatment, right? I know she's done in Mexico and in Panama, and she is her child has other issues as well besides autism.
Okay. So I would just say everyone's situation is unique. You have to do your research and figure it out, but generally the research is still undergoing, so you got to carefully consider your pros and cons.
Okay. I am going to add Daniel to the screen here, and we'll ask him to unmute his mic.
Hello.
There you go. Hi. Are you ready?
I'm ready. Yes.
Okay, we're going to ask Nuwan to stay around a little longer if he can, and we're going to give you the stage now so you can go on to the neuroscience part of it. All right.
Sounds
thank you,
good.
everybody. Around. I am fascinated. Thank you. All right, Daniel, you are up.
Okay, can you just let me know if you can see my screen?
Yeah, I'm adding it to the screen now and then removing.
So we're good? Okay, wonderful. So thanks, Nuwan, for that. I think it was really informative, and I think it builds really well for the next level of analysis, which is where my presentation will come in, which is talking about the neuroscience side of things. So I think Nuwan laid a really good foundation of talking about the genetics, and now what we're going to do is explore post-mortem human brain research in order to understand, what are the impacts that these mutations have on the biology of the brain, basically. So I'd like to thank Autism Belize for having me speak today. I'm always excited to talk about neuroscience and always especially excited to talk about post-mortem human brain research, especially in the context of neurodevelopmental and psychiatric disorders. So like I said, we're going to sort of shift up our approach now and not talk about animal models or stem cells like Nuwan covered, but instead jump into the realm of post-mortem brain research. So there are a few brain banks around the world. Brain banks typically are concentrated on specific pathologies. So you might have a brain bank that is interested in depression, and so they have brains of individuals who died with depression. Brain banks for individuals who suffered with autism, brain banks for individuals with motor neuron related diseases. But what brain banks really do for us is they provide us with tissue samples that can give us a little snapshot into the biology of these processes or the biology of the brain. The great thing about brain banks and brain samples is that these are samples from an individual who was living with some form of pathology. So it's ecological validity, how much it represents the actual pathology is really high because it is the brain of the individual who suffered with those illnesses. So my research is specifically interested in postmortem brain samples and using those to understand psychiatric disorders. But something that I'll talk about later is that there's a really strong comorbidity between neurodevelopmental disorders and psychiatric disorders. And that might have something to do with their shared biology, their shared neuroscience. Now, in order to actually do this research, right, in order to characterize a brain, what we also have to do is to understand the diagnosis of the individual. We have to understand the life experiences that these individuals had. And so the way that you do this is through a method known as the psychological autopsy method. So just like you can do an autopsy on your body, you could also do an autopsy on your mind. And so researchers spend a lot of time trying to understand, how does this diagnosis, how does this life experience, how does maybe even drug use relate to changes in your brain's biology? I know tons of postmortem scientists who each study something really interesting. One of my my fellow researchers looks at the effects of cocaine on the brain through postmortem brain samples. Some of my colleagues work on using postmortem brain samples to understand autism or understand the impacts of severe childhood abuse. And so really postmortem brain samples provide us with this unique opportunity to look at these diagnoses, these pathologies, and explore them on a brain level. So I wanted to make sure that we're all caught up with respect to basic neuroscience. I think there's a lot of times when I'll talk about neuroscience and I'll forget the fact that I'm not speaking to neuroscientists. So I want to orient us towards some really interesting brain areas, and then I'll ground our conversation in how these brain areas and brain networks are impaired in neurodevelopmental disorders, but then also impaired in psychiatric illnesses as well. So I think one of the most beautiful systems of brain regions is the frontal cortex. And the frontal cortex is composed of the dorsal lateral prefrontal cortex, the anterior cingulate cortex, but really without all those complex names, what we can say is that these brain areas are super important for decision-making, executive function, problem solving, language. These are really the brain areas that differ in evolution between us and other organisms. There are also important brain structures beyond the frontal lobes. I think I'm biased because I work on the frontal lobes, so I think they're sort of the coolest part of the brain. But that's the limbic system. So anytime you think about those basic emotions, those basic abilities that our brain is able to accomplish, we like to think about the limbic system. So we know that the amygdala is involved in emotion processing and the experience of fear. The hippocampus is important for building memory networks, often a brain area that is impacted in epilepsy, also in dementias. And so the limbic system is very important for a lot of the emotions that we experience. And I'll connect this back in a little moment. And then, of course, the temporal lobes as well. And these are this is a brain region that's important for both language, but then also hearing and also cognition in some ways. So now that we have a basic understanding of brain structures, I want to go down to the cellular level and talk a little bit about the different cell types that we can see in the brain. And then our conversation will move on and look at how each of these individual cell types are actually impacted in neurodevelopmental and psychiatric illnesses. So, of course, I'm sure that we've all heard of neurons. These are the cells that are, building connections in our brains. They're the cells that are doing a lot of work to communicate signals across different brain areas. And, they're important for the experience of consciousness. They're important for how we're able to lift up our hand. They're important for so many processes. And what we know about neurons is that they form connections with other neurons, and we call that a synaptic connection or a synapse. And these connections are really important for mediating the communication between different brain cells. Now, an entire sort of other class of brain cells are our glial cells. And for a very long time, neuroscientists really ignored the importance of these cells, right? We would always chalk up these cells to they're involved in supporting neurons, right? They're just the support cells. And what we realize today is that this is not necessarily the case. And we're starting to realize that many neurological, psychiatric, neurodevelopmental disorders have something to do with these additional cells that are not neurons. And I'll talk about them in a little moment, but as a cursory glance, we have microglia, oligodendrocytes, and astrocytes. And like I said, we'll come back and look at the pathology in just a little moment. So what we can do as post-mortem neuroscientists is to take a chunk of tissue from whatever brain region that you're interested in and try to understand how is it that, suffering from a neurodevelopmental disorder or a psychiatric disorder changes the molecular biology of the brain. Now, the thing about molecular biology is that it is a very complicated field. Historically, researchers would, ask about a single gene and say, we know that this one gene is linked to this one disorder. Let's investigate that one individual gene. The problem with that, though, is, as Nuwan correctly pointed out earlier on, is that many of these disorders are polygenic, right? They involve so many different biological pathways. And so now scientists use methods that allow us to look at the entire picture. What's going on in its entirety, as opposed to focusing on very specific genes, very specific sites. So one of the signals that we often do look at is RNA and proteins. So what Nuwan was talking about earlier was mutations in DNA. But the thing about those mutations in DNA is that they impact the messenger molecule that we'll call RNA and inevitably impact protein, and those proteins then impact the structure and function of neurons. So we can use technologies to be able to profile a genome-wide type of approach, to look at all of the RNAs, to look at all of the changes in DNA, to look at all of the changes in proteins, as opposed to focusing in on one very specific gene or one very specific protein. And what this allows us is, it allows us the ability to discover new biological pathways that we've not known of before in the context of psychiatric disorders, neurological disorders, or neurodevelopmental disorders. And what's even more interesting is to add to this complexity of it's not just one gene, but rather many different genes. We also know that our environment can have a very big impact on the expression of our genes and can have a big impact on how our cells actually function. So just to put things into context for you, to talk about the complexity of what we're dealing with here, within our genome, we have 3.2 billion base pairs. So 3.2 pairs of those letters that Nuwan was talking about earlier. And then if we look in the brain, we have approximately 100 billion neurons. The estimates range depending on which manuscript you read, that make 100 trillion connections. I can't even imagine what 100 billion looks like. So just imagine the complexity that we're dealing with when we study neurological, neuropsychiatric, psychiatric, or neurodevelopmental disorders. And so by using approaches where we can look at all of the processes at once as opposed to one specific gene can help with discovery and understanding, what are the biological processes that are impaired in a lot of these different disorders. And so, like Nuwan said earlier on, many of these disorders are polygenic and involve many different biological pathways, many different genes that are involved. And of course, there are some exceptions if you're talking about neurodevelopmental disorders, which are monogenic and they involve one particular gene. But for the most part, when we talk about neurological, neuropsychiatric, psychiatric, or neurodevelopmental disorders, many of them have this polygenic component to them. So, despite the fact that the picture is kind of complicated and it's a complex puzzle that we're still trying to make progress on today, we have learned a few things about the biological processes that underlie neurodevelopmental disorders, psychiatric disorders, and so on and so forth. So, I think one of the most interesting hypotheses with respect to neurodevelopmental disorders is rooted in brain development, right? So just to talk about neurotypical development for a moment, what happens in neurotypical development is that our brain has the division and multiplication of cells that will end up becoming neurons. Once those neurons actually form, they begin to form synapses and connections to other neurons. But as we age, as we progress through neurodevelopment, there's a pruning process, right? And that pruning process is where our brain is refining those connections, right? It's almost as if we overshot, built too many connections, built too many neurons, and now what we need to do is refine it down a bit so that we only have the precise connections that we actually need. And this is a normal brain process that all of our brains go through during neurodevelopment. And this is a neurotypical process. Now, some of the hypotheses underlying autism and other neurodevelopmental disorders speak to the fact that this pruning process might actually be impaired. And so speaking to the fact that these connections are not necessarily refined, and there may actually be too many or too little of them. So when we look at postmortem brain research, some of the studies have found specific changes in the frontal lobes and the temporal lobes. And I think what's really cool about this is that it speaks to a lot of the symptoms and comorbidities that you see in the context of autism and psychiatric illnesses, right? The frontal lobes, like I said earlier on, have something to do with planning, decision-making, and problem-solving, and language, and complex social skills, and all of these really important executive functions that humans evolved during evolution. And so it makes sense that one of the sites where we see impairment would be in these really beautiful brain areas. And then, of course, the temporal lobes as well. What's also very interesting is that, when we look at the brain, these different brain structures are all important for different functions. And so you'd imagine that biologically, they also look different at the molecular level. And some research has actually shown that in the context of autism, these brain areas are not as different as they should be. So it's almost like, the frontal lobes are not as different from the temporal lobes as they should be. And we see less of this differentiation between the brain structures. Now, as a result of this hypothesis that, maybe there are too many connections and something to do with the pruning process, what this ends up meaning is that neurons are not able to do their typical job of neural communication, right? They're not able to send out messages to other neurons in order to allow for proper brain functioning. And what's interesting is that molecular research has shown that some of the genes and some of the molecules that are changed in the brains of individuals who suffered with autism have something to do with the synapse. And they're genes that are important in that communication between brain cells. So if we sort of add on to the picture that we have then, what we see is impairments in the refinement of connections, impairments in the number of connections that we have, but then also just impairments in the biology of these connections and looking at the actual synapse and asking whether or not we see typical neural communication. Right, so that's sort of what I wanted to talk about with respect to the changes that we see in neurons. And when we talk about neurons, we can talk about different types of neurons as well, right? The picture is a lot more complicated than what I have here on the slide. But some researchers actually showed that very specific neurons within the prefrontal cortex, those brain areas, that brain structure that's involved in executive function, very specific neurons are actually impaired. So if we look at the prefrontal cortex, it's divided into different layers. And within these layers, you have different organization where certain neurons exist and other ones don't. And in this one particular study that I'm thinking of, the neurons that seemed to be different in autism or in the brains of individuals who suffered from autism were those in layers 2, 3, and 4 of the frontal cortex. Now, just to orient you as to what cells within these layers actually do, these are the neurons that receive information, right? So these are going to be the neurons that take information from all around the brain. Then they're going to communicate that information to the deeper layer, layer 5 and 6 pyramidal neurons, which then they can send out information to other brain areas and regulate other brain areas. Remember that the frontal lobes are so important for executive function, but also important for regulating brain areas that are involved in anxiety, and brain areas that are involved in mood, and brain areas that are involved in so many important psychological processes. And so now we're beginning to build a bit more of a picture and understand that these biological impairments that we see in the prefrontal cortex of individuals who suffered with autism might explain some of the comorbidities that we see with autism, like anxiety or OCD, et cetera. And I'll say more about that in just a moment. Now, I think the next obvious question is, why is it that these connections are impaired? And where there has been quite substantial efforts is trying to understand, why is it that the pruning process is impaired in autism? Why is it that these connections are impaired? And some research has pointed to microglia. So remember, microglia are part of those other cells that neuroscientists ignored for a very long time. And what you can actually see in this image here is that microglia associate with neurons. And they might actually have an important role in responding to inflammation in the brain, but then also regulating synapses, regulating connections between neurons. So the hypothesis then here is that in the brain of an individual who suffered with autism, these microglia may not be doing their job of refining the connections and making sure that only the best connections remain during neurodevelopment. So I think I really love this picture here because it shows you, you have this microglia, which is supposed to snip at specific locations and to make sure that the only connections that we have are ones that we really, really want. And so there might be some type of impairment that happens there between that snipping process and microglia actually doing that job. And of course, there are multiple factors as to why microglia might be impaired. This can include environmental factors like stress, diet, but then also genetic factors as well, or even in utero environment. And what I think you can appreciate from this slide is that microglia are involved in tons of processes, right? Impaired microglia is related to developmental disorders like autism, schizophrenia, Rett syndrome, but then also related to psychiatric disorders as well, like psychosis or obsessive compulsive disorder, or anxiety, depression, or even dementia. So sort of continuing on with our story here, what we're seeing is that, maybe the comorbidities that we're seeing has something to do with a linkage in the biology that we're seeing that underlies autism. So the last cell that I want to talk about are our oligodendrocytes. And what oligodendrocytes do, I think they look like octopi, but what oligodendrocytes do is that they enwrap neurons so that neurons can be more efficient at sending electrical signals that end up leading to communication with other neurons. And so what the research has shown, postmortem brain research, is that oligodendrocytes might also be impaired. And this impairment will lead to less efficient communication between neurons within different brain areas that we've talked about so far. So just to wrap up, I know that my part was a little bit shorter than Nuwan's. What I'd like you to appreciate here is that when we think about the neuroscience and neurobiology of psychiatric illnesses and neurological illnesses and neuropsychiatric illnesses, we're often talking about similar pathways, right? And that shared molecular biology might somehow help to explain some of the comorbidities that we see amongst all of these illnesses, right? So if you think about depression, the ugly twin sister of depression is anxiety, and vice versa. When you think about autism, you often see comorbidities with obsessive compulsive disorder, or sometimes comorbidities with seizures. And there has actually been some work that's shown that there are molecular bridges, right? That they might share something similar in their biology. And in fact, today, one of the sayings that we often use in the field is that comorbidity is the rule and not the exception, right? It's actually the exception to just have one pathology, and it's instead the rule that many of these pathologies coexist. And that's just because of the fact that our brain is so complicated. We can't impair the prefrontal cortex and these important brain areas that are involved in executive function without having an impact on a variety of behaviors, cognitions, moods, emotions, etc. So that's sort of the direction I wanted my conversation to go in today, to equip you with some of the terminology that we use in postmortem neuroscience so that you can use that now to investigate also on your own and learn about, what's happening with microglia, what's happening with oligodendrocytes, what's happening with neurons in the context of autism, but then also to be aware that these comorbidities exist and there might be a biological explanation as to why this is actually the case. Okay, so I will go ahead and take any questions if any of you have them, and thank you for having me.
Okay, so we've got everybody here. Let's see, I don't know if we have any questions yet. Anybody out there that wants to ask anything? So, the synapses are firing too fast. We've got this issue. What do we do? Is that what you're trying to figure out now? Is there any recommendations for us parents out here?
Yeah, I think that's a really interesting question. I think in the context of my research, I'm really interested in sort of life experiences and how they increase our risk for different psychiatric illnesses. And so the question would be, what are the time periods in which the brain is able to recover from an environmental life experience? And I think that's where a lot of the research is looking at developmental windows and when can we rescue these pathologies.
Sorry.
Hi, Mateo.
I know, right? He's a TV hound, just like his mother. He likes the attention. Go figure. Must be something in the biology there too, huh?
Maybe.
We have something here from a parent. You mentioned the difference in frontal lobe and temporal lobe. Can you say more?
Yeah, absolutely. So just talking about location of where they are. So the frontal lobes are the most rostral aspect of our brain. These are the frontal lobes actually continue to develop and refine themselves all the way up until 29 years old. And what's so interesting about the frontal lobes is that our understanding of frontal lobe development explains why sometimes teenagers make really bad decisions. Their frontal lobes are not fully developed at this point. Describes why a teenager, goes and takes the car when they don't have a license. So the frontal lobes have this protracted development where they're continuing all the way up to 29 years old, and are really, really important for executive function, decision making, problem solving. Whereas when we go to the temporal lobes, so on the sides here of our brain, the temporal lobes have really important relationships with memory, right? So you've probably heard of frontal temporal lobe dementias, and that's where the pathology of various dementias starts within the temporal lobes. And so the temporal lobes have a role more so in memory, in hearing. They're also involved in the understanding of language, whereas the frontal lobes tend to be involved in the production of language. So that would be Wernicke's and Broca's area. And so those areas are often correlated with aphasias and language impairments and so on and so forth. Hopefully that speaks to...
Here, because you're telling me the frontal lobe develops until 29, and that has to do with the production of language. So my son, who is still nonverbal, so I've still got a chance to keep working at him because that keeps developing until he's 29. So I shouldn't give up on speed.
Yeah, I think I'm a huge advocate of neuroplasticity and the idea that our brain can change in response to experiences. I think what's always really important is to understand that, yes, while our brain can change, that change might not be the same as someone else's brain, right? And so, it's about improving and developing that neuroplasticity, but that neuroplasticity might not bring us to, a level that would be similar to someone else that's age matched and isn't suffering with some type of neurodevelopmental or psychiatric illness.
Another question here. Are there differences in these areas of the brain in autism?
Yeah, absolutely. So even just looking at the molecular biology of these two brain areas, typically they're different, right? Because they do different functions. But in autism, they're a little bit less different from one another.
Slide, it looks like you're saying there really should be more different.
Exactly, yeah. And also, a lot of the research has looked at synaptic structure, so the ability of neurons to wire together and build networks together as being impaired in these brain areas as well.
Question for Nuwan.
Okay.
Hi. Yes, somebody messaged me and asked me about there's those DNA tests. Just to be clear, sending away for one of these DNA tests that was advertised online, that's not, will that give us anything? That was the kind of testing you were talking about when you're talking about genetics.
No, generally testing would be run through a hospital or a clinician. I wouldn't recommend for this purposes to go through something like 23andMe or anything like that.
Okay, just to be clear, because somebody messaged and asked me about that, but that would be different. Okay. Any other questions? Any recommendations for us as parents who are working with their children, trying to decide what therapy, what medication, whatever? There's something that we I know you said you're always looking for people who need us, our kids, our blood, or things to study. Anything else that you need help with research along, or that you could recommend for us? Still not in that stage.
I guess I'll take a crack at that first. At least for parents of children with the type of disorders that I've discussed or researched, so these severe, rare, monogenic disorders, at least for researchers, it's very helpful for us if parents get involved with these communities that are tailored around the specific mutations or syndromes that their child might have. For example, I'm studying a disease called FOXP1 syndrome, and there's a whole foundation that is associated with parents and researchers who are studying and who care about this rare disorder. So getting involved with foundations or organizations like that is very helpful because you're able to learn more about the disorder, more willing and able to participate in studies, which obviously helps us a lot. So we're a little unique in that we have children to give us their urine or skin cells or something. So that's also quite helpful for us, obviously, so we can study those cell types. But at least for research, that's mostly beneficial for us in terms of helping us advance our research.
Daniel, for you, anything you would recommend for us to do to help with the research?
Yeah, so I think, speaking back to the idea of plasticity, there's tons of work to show that a variety of things can lead to the remodeling of our brain. So exercise, healthy eating, etc. And again, the idea is that, while our brain is plastic, let me say that again. While our brain is plastic, I think it's important to understand that all of our brains are different. And so we might respond differently to environmental stimuli than someone else's brain.
Have you guys ever heard of the Anat-Daniel method, the awakening of the brain? Ignore the thing there.
Huh.
They talk about brain neuroplasticity and they use slow movements and stuff. So if you haven't before, that's my recommendation to you guys. But you might want to take a look into this.
That's really fascinating.
Okay, taking note.
It's one of the new therapies that are out there. It's mostly in the U.S. We have one person who before COVID had started coming down to work with kids here. But it's about that. It's about kind of helping you.
Yeah, I think that's really cool. So now that we have that fundamental understanding of neuroplasticity, to, maybe if you're interested, to see whether or not the therapy that you're pursuing has been shown to change the brain before, and to look into the literature to actually explore that.
Guys, I am so very, very... Ooh, hold on. We've got something here. I'm going to just put it on the screen. Just to add for parents, while it certainly does not address the genetic answers like they were being discussed, chromosome microarray or whole exome sequencing test with you. A test like 23andMe can help inform supplemental research under the direction of a trained functional medicine integrative med doctor, which are often very helpful. Wonder if we can find a doctor. Something else for me to look into. Thanks for giving more work. Okay. Thank you for that, though, because someone immediately messaged me and asked about this. I think that's it. Any last questions before I let these guys go? All right. Thank you guys so much for doing this. And give it. I know we're lay people, and you all really helped break it down. So I was able to call for post-mortem. I might have to pronounce all the words, or I now know where the parts of the brain are from the thing. So, on a serious note, thank you so much for giving us this information. We have 50 million questions on new questions every single day when we watch our kids take part in our behavior, why they do or don't. So just getting a little bit of background helps. And it also helps us to know that people are working on it, that they can help us. Maybe in the future, help us manage our medication, help with behaviors, and not even just with us. So thank you so much for taking the time. Help us. I know you've had several thank yous as they've gone along that have come up. The comments. So I know everyone is really grateful. We will be saving this broadcast and putting it up on our website. So at any given time, a year from now, whatever everybody else is, watch again, encourage others to watch. It will be there at autism.org. Thank you guys very, very much. Have a great evening.
Thanks for having us.
Okay. Bye.
See ya.
And for all of you who stuck around, this is probably our longest presentation. I hope you did enjoy. It was different than what we've done before. But I really am grateful for these young men for what they're doing and their research and for taking the time to come. See another comment here from San Pedro. Let's see. Awesome. Very interesting and educational. Thank you from San Pedro. Amberg is key. So maybe you guys should think about coming down and visiting. I think there'd be several people around that'd be happy to be tour guides. Once COVID and everything is all settled up there for you guys. I know you all are still locked down. But thank you very, very much for joining and listening and learning with me today. We will be having another presentation in July that we're working on. It might be slightly different than just a live. I think we want to do more hands-on, one-on-one training with parents, but I will be announcing more information on that by next week. If you have any other questions or concerns or anything, you can post them here, but you know you can always reach me on WhatsApp if you go to the website or on Facebook, and I will answer. I hope you guys are doing good. For those of you who missed our parents meeting last weekend, it was fabulous. I met some awesome parents. We had a great time together. Friendship engaging, lots of ideas, a way forward. I love the energy. So I really hope that you all will join us for our next parents meeting. It will be the last Saturday of every month at the YWCA. So you can just pencil it in your calendar, because it will be coming up again for the last Saturday of the month, every single month at the YWCA Belize. Thanks again. Have a great evening, and I will talk to you guys shortly. Bye-bye.